Welcome everyone to our fifth session. We'll get started in just a minute as we wait for people to come in and finish up with the poster session. All right, I think we're going to get started with this, our fifth short talk session of the symposium and our final one, certainly not the least. For this, we have three excellent presenters who will be talking about some more work on computational and structural approaches to GPCR research. We were fortunate to get lots of great abstract submissions on these topics, so we have this additional session, and in order to keep to time, I'm going to get started right away. Our first speaker for this session will be Luca Chiesa of the University of Strasbourg, who will be talking about LIT-AlphaFold, which is a new tool for multi-state modeling. Thank you, Luca, and feel free to share your screen and get started.
Okay, do you all see my screen now?
Yep, looks good.
Okay, perfect. So thank you very much for the organizer for allowing me to present you our latest work on how to use AlphaFold to model alternative state conformation of proteins. So, sorry. Okay, so for those of you who do not know what it is, AlphaFold is a deep learning tool for the prediction of the 3D structure of a protein starting from its amino acid sequence. Since the first release in 2020, AlphaFold has become the current state-of-the-art method for both prediction of monomeric structure of proteins and of protein-protein complexes using AlphaFold multimer, and now, since a few months ago, also for the prediction of every biomolecular complexes using AlphaFold 3. But now the question is, why am I talking about AlphaFold at a GPCR meeting? The reason is that despite the great results of structural biologists in the last few years, there are still many targets in the GPCR family for which we do not have access to an experimental structure. Or if you want to be even more focused, there are many complexes between GPCRs and other proteins for which we do not have an experimental structure. And at the moment, AlphaFold is the best tool to bridge this gap, since it can generate easily and in a quick way good quality model for this structure. However, there is a catch, and that AlphaFold can predict only one conformational state for each protein. And this is a big deal for GPCR research because, as you all know, GPCRs have at least two main conformational states, an active state conformation and an inactive state conformation. So we would like to be able to model both of these conformation using this tool. Lucky for us, in the last couple of years, different methods have been developed to do so. And despite the differences between them, they are all based on the same philosophy, that you can change the output of AlphaFold if you change the input information that you're giving. So you can get a different conformation if you change the information on which this conformation is predicted on. Which now you might ask yourself, okay, we cannot change the amino acid sequence we give as input to AlphaFold. How can we change the input? In reality, AlphaFold is not as streamlined and one might think. In fact, when you give your input sequence, this is not used directly to predict the structure, but first it is searched in genetic databases to find other similar sequences, which are then aligned in order to form a multiple sequence alignment from which coevolutionary information are extracted. Then the same sequence is searched in the protein databank to find similar proteins for which we have an experimental structure and we can use as templates for prediction. So the final 3D structure that you obtain from AlphaFold is a product of your input sequence, the MSA that was identified, and the available structure templates. So if you can change the MSA and you can change the templates that you use for prediction, you can have some form of control over the conformation you are predicting. To give an example of this, the oldest of this method is AlphaFold multistate, which some of you might have already seen or at least used these models. In fact, using it, it's possible to model both the active state and inactive state conformation of GPCRs. This is achieved by filtering which templates are used for prediction. So if you want to model an active state structure, you will only select active state templates or inactive state template for the opposite case. Then once you have selected which structure you want to use as reference, you have to change the sequence information by masking the position in the MSA, which are already covered by the templates. So in this way, the prediction will be more based on this biased information regarding the activation state rather than on the more obscure coevolutionary data. Other than AlphaFold multistates, there are many other tools one can use to model alternative conformation or to perform conformational sampling using AlphaFold, each one with its own upside and downside. So ideally, in a modeling project, you should test how each method performs in the prediction task. The problem is that each pipeline has been developed separately from each other. So if you want to test more than one method, you will have to spend a lot of time just installing programs and harmonizing the input information, how the input information is processed and how data are handled. And this can take away a lot of time that you will more likely spend in more interesting research. So we decided to try and streamline this process by developing our own pipeline, which we called LIT-AlphaFold, which groups all of these tools into a single place and streamlines the entire process. So to not have just a simple end-to-end prediction pipeline, but splitting it in 3 steps: input preparation, input modification, and structure prediction. So in the first step, the sequence and template information are searched either in local databases or using the online web server from CollabFold. Then, once you have all this information, these are saved in individual files from which when you can change the data you are using. For example, you can filter the templates you have selected based on their PDB ID. Maybe you have some structure that you will really like to use for modeling or structure that you really don't want to use for modeling. Or you can filter them based on GPCRDB annotation. So for example, you want active state structure, but you don't want to search for all the active state structure. You can just tell the pipeline that you want structures that are annotated as active, and automatically they will be filtered. Then once you have selected the templates, you can modify the MSA by removing it, clustering on by performing in silico mutation. And then once you have prepared your input, you can then run prediction. The pipeline does not only run prediction, but lets you change some of the parameters. So for example, you can set how many sequences you want to use for your prediction. You want to use the full MSA, you want to use a smaller part of the MSA. You can decide how many times you want to repeat the calculation, since there is a random component in the prediction. And also we allow for other parameters. The pipeline is built to handle all types of predictions, so you can predict monomeric structure, homomers, heteromers, and we also offer support for large-scale dimer studies. To give an idea of what this pipeline can do and how you could use it, we have recently used it in a study, in a benchmarking study, evaluating the quality of chemokine receptor complexes predicted by AlphaFold. So chemokine receptor are a family of class A GPCRs that bind chemokine, which are small soluble proteins that bind in the extracellular pocket. And in the study, we evaluated how well AlphaFold can reproduce these complexes, looking both at the overall quality of the complex, specific interaction hotspot, and what I will present to you is the activation state of the receptor structure, since we're talking about multi-state modeling. So what we observed first is that if you run your calculation at default values, for half of the receptor we studied, we obtain only structure in an active state conformation, which makes sense because most of the chemokine we analyzed are agonists of these receptors. One of them is in an inactive state, for which we use an antagonist chemokine, and for 4 of them, we obtain multiple activation states. So as you can see, for example, in the picture, for CCR5, we obtain both active state structure of the receptor and inactive state structure of the receptor. So, which was not ideal, because you would like to have only active state structure or only inactive state structure of your receptor. So we try to modify a bit the information we're giving to AlphaFold. So for example, we filtered the templates, selecting only active state templates or only inactive state templates. In this way, we're able to selectively model only one conformation of the receptor, for example, only the active state or only the inactive state. For other targets, it was more complex because just template selection was sufficient to model both conformation state only for 5 receptors out of 9. We were able to model the active state of another receptor, 6R4, by modifying the MSA, removing a part of it, other than selecting the templates. And this was a great achievement because we were able to force a unfavorable conformation for this dimer. And finally, for the best results were obtained by modifying directly how AlphaFold works by applying dropout, which is a deep learning technique that is used to obtain more diverse prediction from a model without affecting its accuracy. Now, the most interesting thing we observed from this study was that regardless of which method we used, the quality of the protein-protein interface that was predicted was still of high quality. Here are shown the DocQ values, which DocQ is a measure for structure prediction quality. And as you can see here, we don't get worse performances by changing our settings. So you can still get good performances at the interface while still having the correct activation state. So in conclusion, we developed LIT-AlphaFold, which is a new input preparation and structure prediction pipeline for AlphaFold, which allows you to, gives you the tool to model alternative conformation of proteins by having more access to the information they're used for the prediction. The code is publicly available on GitHub. You can scan the QR code here, or you can go to this link. And here we not only give you the code, but we also have a series of tutorials on how to use this tool for your modeling needs. The pipeline has been tested to predict both monomer structure, and by using it for the first time, we were able to model a protein, an alternative protein conformation in a dimeric complex rather than just in a monomeric complex. So finally, I would like to thank all the members of my lab, the Laboratoire d'Innovation Thérapeutique at the University of Strasbourg. In bold are the people that directly participated in the study, and I would like to in particular to thank Lauri Urbas, who is another PhD student in the lab, who ran most of the benchmarking studies on chemical and chemokine receptors. And I would like to thank all of you for your kind attention.
Thank you, Luca, for the nice talk. I would like to ask the audience if they have any question. We have time for one quick question. If that's not the case, I can go ahead and ask one. Actually, it wasn't very clear for me because you mentioned that in the end, filtering MSA improved the result better, right? And yeah, because I mean, it's not clear how MSA over filtering the template structures, you know, could result in this in the improvement of model, because MSA technically you basically modify the like the not the structure but mainly like the input for so it's not clear why not the template modification and instead MSA. Could you please comment on that?
Yes, because unfortunately I did not have the time to go into the details on how AlphaFold work. The thing is that AlphaFold mainly works out of coevolutionary data. So it's not intuitive, but the 3D structure is not predicted from the template, it's predicted from the sequences. So it's the same reason why with AlphaFold multistate, you don't just filter the templates, but you also remove part of the sequence. Because if you don't remove the sequence, filtering the templates is absolutely useless. It doesn't change anything. What we saw is that for multimer, you don't need to be so drastic. You can keep more sequence data. And even without removing sequence data, you can still kind of force a specific conformation just by filtering templates. For certain targets, it's more difficult for many reasons. Maybe it's because of how the model was trained, or maybe the templates we are giving are not good enough. They're not similar enough to force the model to accept them. So we show that if we reduce the number of sequence that we use for prediction, we are able to kind of force this alternative conformation that otherwise will not be sampled in an easy way.
Yeah, okay. Makes sense, definitely. Thanks a lot. I don't see any more question. If there is any question, please just copy them, put them in the chat, and Luca, I'm sure, will take care of that. Thanks.
Thank you.
Thanks so much, Luca. We really appreciate your interesting talk. At this point, we'll be moving on to our next talk, and that is by Maria Giovanna E Papadopoulos, who will be discussing work on the discovery and characterization of small molecule TGR5 ligands with agonistic activity. So thank you. Go ahead, Giovanna.
Yeah, thank you very much for the kind introduction. Let me know if you can see full screen.
Yes, looks good.
Perfect. Yeah, so today I would like to give you an overview of the recently published paper discovering characterization of small molecule TGR5 ligands with agonistic activity that was published 2 months ago. And I also would like to give you a few insights also in the background story of the study. So yes, the study, first of all, I would like to introduce the target. So TGR5 is a bile acid GPCR. So it is coupled to a G-alpha stimulatory protein, which when activates the adenylyl cyclase leads to an intracellular accumulation of CMP. And it is activated by the bile acids, which are formed in the liver by the oxidation of cholesterol. So this receptor is actually ubiquitously expressed throughout the body. And what the results of these activations is the energy expenditure, insulin secretion, and the reduction in the inflammation. And this is the main reason why this target is very interesting targets for metabolic disorders and also for inflammatory diseases that can also have an influence in metabolic disorders, particularly diabetes type 2. So this study started during my thesis, my master's thesis, where AlphaFold was not available at the time. So I created a homology model of the active state of the receptor, and we screened the zinc-15 lead-like library. And what we found after we tested those compounds was 4 compounds that have a positive allosteric modulator effect. So as soon as the structural data became available, we wanted to go back to this project and figure out where these compounds go and bind. And you can see in these 2 published structures that there is an orthosteric ligand here, P395, that binds to the orthosteric pocket, but you can also see a ligand, the cholesterol, in the cholesterol binding pocket, very commonly known as. But in the second structure, they solved the semi-synthetic steroid INT777 in both the orthosteric and the allosteric side. So you would guess that the PAMs, well, we would guess that the PAMs would go and bind to this cholesterol side, but we wanted still to do the pocket exploration, and we used 2 different programs just for the location of these compounds, and one was SiteFinder from Molecular Operating Environment, the other one was an online tool, SwissDoc. And you can see the first image here, I have color-coded darker green is the highest ranked and lighter green is the lowest ranked pockets that were found from MOE. And it was actually the highest ranked pocket in both cases, in both SwissDoc and SiteFinder. And we were also able to identify this cholesterol pocket, but it was not so highly ranked in both programs. And to make this W75 pocket, I will call from now on, more interesting, there was this other cryo-EM publication that the resolution was not great, so we did not use it. However, they had a claim on this W75 that could be a lead for the orthosteric site, but did not have any effect on the agonist when mutated to an alanine. So for that reason, we decided to proceed with MD simulations to explore this W75 pocket. Do we see any differences when the tryptophan is there or when we mutate it to an alanine? And when we created the control systems, you can see here a GIF that shows that the orthosteric ligand in green P395 is actually not completely stable in the pocket. So it kind of changes and moves around. So we decided to take that into account as well and cluster all the frames of the MD simulations and maybe consider this dynamic effect of this receptor for the next screening campaign. So to get more into the analysis of the MD simulations that we did, here you can see the system of the wild type. So in teal, you see the PAM, and in light green, you see the orthosteric ligand. In magenta, you see these W-tryptophan 75. And we calculated the distance of the orthosteric ligand from the center of the receptor, which you can see is quite stable, doesn't change much. And we also calculated the contacts from 0 to 1 of the PAM with the protein. And you can see that at least for the first 200 seconds, it stays quite in contact. Well, when we analyze the mutant with the alanine, you can see that there is no more this hydrophobic core that coordinates the orthosteric side with the allosteric pocket. So you can also see that in some cases, the ligand is not so stable inside the pocket, and the contacts are lost quite fast, like in the first 100 nanoseconds, you can see the ligand that almost flies outside of the pocket. And this led us to do two different approaches on the virtual screening campaigns. One was focused on this W75 pocket, and the other one was focused more on this dynamic process of flexible ligand protein complex. And we used this time a tailored Zinc 20 library, which means that we took into account the log P and the molecular weight of the already known ligands for TGR5. And from the first screening, we found one compound that acted as a PAM, and we decided to use this compound through Small World, which is a similarity search engine that searches through Zinc 20. And we redocked all these compounds and led to 4 hits, 4 agonists and 1 PAM. While with the second approach, we just cluster the frames of the MD simulations with k-means. And what we did was to consider all the compounds from the top 10,000 of these different simulations that we did and visualize only the ones that were present in at least 2 of the dockings. And we came across 3 hits, 1 agonist and 2 PAMs. And we verified this information with in vitro assays, mostly with luciferase and EPAC assays. You can see here, this is the most promising agonist, and we were able to obtain also concentration response curve with an apparent EC50 of 19 micromolar. And we also tested the PAMs. You can see here we tested them not only with endogenous ligand lithocholic acid, but also with the orthosteric ligand that we evaluated our MD simulations with. And lastly, we were able also to obtain a curve that shows that this PAM has an effect on the efficacy of the LCA when binding to the receptor. So with that, we were quite convinced that these ligands were binding to this W75 pocket, but that was not the case. So you can see here that the first image you see this W75 pocket, and in the second you see this cholesterol pocket, and the only polar residues are threonine 131 and serine 123. We saw some interactions when we docked the ligands there, so we decided to check also these residues. And to our surprise, there was no correlation between these mutations to alanines with these residues. So the only information that we were able to take out of this information, of this screening, was actually to kind of separate these 2 sets And we were able to see that the compounds that had more of a PAM effect on the receptor had at least one aromatic system in their structure. And also they had a hydrogen bond donor and a hydrogen bond acceptor, while the agonists tend to have 2 hydrogen bond acceptors. Yes, and with that, I would like to conclude the presentation. So we were able to find 4 novel non-steroidal agonists, which is what we were searching for, and 4 novel non-steroidal PAMs. In literature, most likely you will find steroidal compounds. We were able to distinguish these 2 categories with common features, structure, pharmacophore. And we still want to optimize these hits. But still the question remains, where do these compounds bind? So maybe we are not certain about the amino acids, but maybe the location is could still be one of the pockets that we explored, but we still need to find the specific amino acids that are involved in the activation of the receptor and leading to this positive allosteric modulation. With that, I would like to thank my collaborators, especially Alexander, Verena, Angela, from the University of Vienna that were involved mostly into the experimental parts, Brian and Jana for hosting me in their lab, with the Ernest STSM shorter scientific mission, for all the MD simulations that we did together. And of course, my professor and advisor, Peter Kolb. And of course, all of you for your kind attention. And I'd be happy to get any questions if you have.
Thank you, Giovanna. It was very lovely. Is there any question for Giovanna? Otherwise, I have one. So I was wondering, like, because in the beginning you showed these 2 pockets. Okay, it's good that we have time for another one. I will continue mine and then I will continue with the one in the chat. But like, so you showed these 2 pockets, one orthosteric and one in the cholesterol binding site. Do you think any of your ligand can bind to the second one, like the cholesterol binding pocket, and that's why you couldn't see, for instance, the effect? So like they can choose between these 2? And then that's why you don't see the effect of the mutation and in the functional assays.
The idea was to check both of the pockets as a negative control, because all of the indications, the computational indications, showed that the other pocket was the one where they binded, and none of the 2 were actually showed any kind of correlation with the activity, with the positive allosteric modulation of the receptor. So maybe what can be true is that those ligands can still bind there, but maybe these particular residues are not the ones that, whether they're mutated or not, has no influence on the activation of the receptor. So there's no effect on the functional part of the receptor.
Mm-hmm. Or it might be that like your pocket is big and like one single mutation is not enough for
yes, of course,
seeing the effect.
Yes.
So I see a question from Frank in the chat. Great talk. Is tryptophan 75 conserved across this subfamily of receptors?
There is actually only one receptor in this subfamily, but this is a great question. It would be lovely if there were more receptors, but it's the only steroid receptor and the only bile acid receptor. So we were quite happy to see this structural data come out in 2020. So we had like to check what else can be found through these cryo-EM structures.
Thanks a lot. Thank you very much, Giovanna, and we can continue with our next speaker.
Yes, thank you, Giovanna. That was a really interesting talk. I appreciate that we're getting a lot of coverage of some, understudied receptors that I personally don't know quite so much about, so I'm learning a lot. And we'll be continuing that with Sumit Bandekar from the University of Chicago, who will be talking about his structural studies of the CellSir-1 extracellular region and a compact multi-domain module of 14 domains, which regulates their signaling. So please, Sumit.
Let me just get my screen going. Okay. Okay. Hello, everyone. I'd like to thank the organizers for inviting me. Today, I'm very excited to tell you about my work regarding the Seltzer subfamily of adhesion GPCRs. Hopefully, everyone can see okay. So yeah, I'll start with some background. So adhesion GPCRs are the second largest family of GPCRs, but they are still understudied. There are 33 adhesion GPCRs with diverse multi-domain extracellular regions. They are implicated in developmental disease, neurological disease, and cancers. Adhesion GPCRs are characterized by their large multi-domain extracellular regions, and they typically bind to large protein ligands that are often presented by opposing cell surfaces. And so what's unique about the ECR in this case is that it both has the ability to sense ligand binding, but also communicates this to the 7TM. And so despite the roles and the number of adhesion receptors in their biology, there aren't any FDA-approved drugs targeting these receptors yet. And so we think that this is because the specific molecular details of how the extracellular region senses ligand binding and transmits this information to the 7TM remains unclear. And so our goal is to understand this for the adhesion family of GPCRs. So for this work, we studied the cadherin EGF laminin G 7-pass G-type receptors, or Seltzers. These are highly conserved adhesion GPCRs. They have a very large ECR, which you can see here outlined as a cartoon, with 23 adhesion domains covering about 2,500 residues. So broadly speaking, there is an N-terminal cadherin repeat region here in yellow, and a C-terminal EGF and laminin G region, and this concludes with the conserved adhesion GPCR gain domain. So Seltzers have important physiological and pathological roles. They're largely involved in development, including neural tube closure, lymphatic valve formation, ciliary development. So the connection between these processes is they all involve some kind of force being present. They also are involved in a few pathologies, including neural tube defects, lymphedema-Joubert syndrome, and Tourette syndrome. In regards to their ligands, the best known ligands of Seltzers are themselves. So Seltzers are thought to dimerize and trans to mediate their biological effects. And this is currently thought to occur through the cadherin repeats, which are shown in yellow here. However, as I mentioned previously, the mechanistic details of this process of how the cadherin repeats may dimerize, and overall, again, how the extracellular region senses ligand binding and transmits this into some signaling output remains unclear. And so we studied this for Seltzers using a combination of structural and functional methods, which I'll cover in this talk. So we started by purifying the full ECR, and we performed single particle cryo-EM analysis. It's important to note that these data were collected in the absence of added calcium, which I'll come back to later. And so overall, we were quite surprised that we didn't see beads on a string here. You know, we have 23 domains. And what we saw was actually overall a compact, well-ordered particle. So will the movie play? Okay. So here I'm showing you the final density map of the reconstruction with the model built into that. And so what we observed were that 14 of the 23 domains in the extracellular region, rather than being extended and looking like beads on a string, they actually form this compact module of domains. And this particle is overall, it's kind of triangle shaped and relatively flat in one dimension compared to the other two. So to go into more detail, our structure contains the C-terminal about 60% of the extracellular region, starting at the 9th cadherin repeat, cadherin 9, and circling all the way back around to the purple gain domain here. So to orient you relative to the rest of the receptor, the rest of the ECR and the rest of the receptor, at the top of shown the structure here, we have the N-terminus of the construct, which would contain the rest of the cadherin repeats, which are not resolved. And then on the left here is the connection to the 7TM, which in this case would be, the membrane would be in the plane of the page that I'm showing you. So the longest dimension of this particle is sitting, we think is sitting more or less flat relative to the membrane. And so to clarify, hereafter I will refer to this part of the protein as the cadherin 9 through gain compact module, or just compact module. So the striking feature of this structure is what we found that two domains, which are very far apart, so over 1,000 residues apart in primary sequence, including the yellow cadherin 9 domain and the purple gain domain, they actually come together in the three-dimensional structure in order to stabilize this compact association or module of domains. And this involves a variety of hydrophobic and aromatic residues, just a few of which I've shown here. So now that we learned something about the C-terminal portion of the protein, our next question was, could we get any structural information about the N-terminal cadherin repeats, which we didn't see in our structure? So going back to selected two-dimensional averages from cryo-EM, we actually observed a fuzzy density, which you can see here pointed out, which is linked to the N-terminus of the ordered portion of our structure. And so we reasoned that these might be the remainder of the cadherin repeats, just kind of flopping around in solution. And so we looked into the literature, and it seems to be a common observation that cadherin repeats often rigidify and also dimerize in the presence of calcium. So we then wanted to study this receptor in the presence of calcium to see how that would affect the structure. And so using size exclusion chromatography coupled to multi-angle light scattering experiments, we saw that in comparison to the absence of calcium, shown here in blue, where we observed a monomeric species, in the presence of calcium, we observed a shift to a much larger particle in solution, and also this corresponded with a molecular weight in agreement with a dimer. And so this is consistent with the idea that the cadherin repeats may be mediating a dimer in the presence of calcium. We also use small angle X-ray scattering data to calculate low resolution molecular envelopes of either of these species. And so here you can see the earlier cryo-EM density map overlaid with a bead model that is calculated from the SAX data. And so here we can see that in the absence of calcium, the bead model agrees pretty well in size and shape with the cryo-EM reconstruction. And then in the presence of calcium, what we see is kind of cool. We see a drastic kind of much larger species that is extended in nature. And this species is about 500 angstrom in the longest dimension. And so if we attempt to model this, so in agreement and in addition, when we take into account some other studies that were published this year and last year, in addition with our data, what we think is going on is that we have this anti-parallel dimer between two cell molecules that are potentially opposed on two cell surfaces. And this involves cadherin 1 from one monomer interacting with cadherin 8 of the other monomer, and so forth. And so from here, we wanted to test, we had identified two putative structural modules of the protein. So the extended cadherin repeat module in yellow, and the compact cadherin 9 through gain module in blue. And so we wanted to test what's the role of each of these modules in cell-mediated adhesion and signaling. And so in order to do so, we designed deletion constructs where we basically deleted either the cadherin repeats or the C-terminal compact module that we see in our EM structure. We then tested these using assays first for cell adhesion. So shown here is a cell aggregation assay where we take two cell populations that are transfected with our gene of interest and either RFP or GFP. And we basically will transfect these cells, lift them off the plate, and then we mix them for a while and we see if they form aggregates. And so here we can see in comparison to the empty vector, wild type CELSR1 was able to form aggregates. And so when we take a look at these two deletion constructs, when we deleted the extended cadherin repeats, we lost the cell aggregation activity, shown here in blue here. And then when we deleted the compact module, we saw a small decrease in aggregation activity, although the cells could still aggregate. And so this suggested that the extended cadherin repeat module is the most important part for the CellSur's adhesive activity. We also wanted to test which of these constructs were important for signaling. And so to do this, we used a Brett-based G protein turnover assay. And in this assay, CellSur was previously shown to couple to G-alpha-S. And so basically in this assay, a lower Brett indicates a nucleotide cycling of G-alpha. And so what we saw here is that in yellow, the wild type protein was able to induce lower Brett, suggesting that the wild type protein can turn over G-alpha-S. The deletion of the extended cadherin repeat region also could turn over G-alpha-S. However, the deletion of the compact module, shown here at the top, lost its ability to turn over G proteins, suggesting that the compact module of the protein may be necessary for regulating signaling. And so to put all of our data together, we have come up with a model to integrate all of the observations that we have. So we think the way that CellSur's operate, generally speaking, we have this N-terminal extended cadherin repeat region that performs the adhesive function of CellSur's. And then we have the C-terminal compact cadherin 9 through gain module, which may regulate signaling, perhaps by talking to the 7TM. And so what we propose here, of course, this is a little bit of speculation. We propose that these two events are linked, as this domain architecture is conserved as far as C. elegans. Furthermore, CellSur's are present in many physiological roles where force is present or is applied. Some examples of this are the lymphatic valve or the neuroepithelium, I believe. And so what we think is going on is that force application may induce some conformational changes. In a more drastic sense, perhaps it could even induce the partial unfolding or unraveling of this compact module. And then this may ultimately result in a different signaling outcome. And so although we still have a few questions, we still have quite a few questions left to answer in regards to this receptor, we do provide some mechanistic framework to study CellSur's with and to understand them better. And then finally, I'll say that we have provided some pathways and some groundwork to maybe design some modulating compounds that could potentially affect the steps of this process. And so with that, I'd like to thank you for your attention. I'd like to thank the members of our lab, including Ethan and Shimon, who were directly involved in this project, our EM facility, our collaborators at Vanderbilt, the Sando lab, our SACS facility, and our funding sources. And with that, I'll be happy to take questions.
Thank you, Sumit, for the nice talk. We have already one question in the chat from Saeed. And he says, have you tried mimicking peptides to block the two monomers interacting?
Okay. Yes, so that's a great question. The big problem right now is that we don't know how, the best thing that we have is we have this SACS data, which doesn't show very high resolution, obviously. And then I can show you some data that's from another group and so this is using CellSur2, where they show basically here again, the way that we interpret this is that we have this extended region in the middle, the compact region on each end, which corresponds to what I see in my EM structure. And so what we think is happening is that these cadherin repeats are making this, dimer where they interact in this anti-parallel mode. But unfortunately, we don't have, at the atomic level, we don't know what's going on. So we need to either try AlphaFold or somebody needs to do a structure of that so we can learn which specific residues are interacting. And then, of course, after that, it would be really cool to design something like what you suggested. Thank you for the question.
Thank you. for presenting. And I think with that, we can already conclude the session. Thanks again to all the excellent speakers.
Yes.